Hello everyone, I am Young Song, a researcher at Ilias Biologics. Ilias is a biotech company based in South Korea developing therapeutic exosomes. In this brief presentation, I would like to introduce our review paper on currently available exosome platform technologies and the human exosome producing cells for the development of therapeutic exosomes. Exosomes are generated from the endosome and released by fusion of a multivascular body and cellular membrane. The released exosomes are taken up by the recipient cells via various routes such as endocytosis, phagocytosis, leaf-left macropinocytosis, and direct fusion, but the exact uptake mechanism is unclear. After being taken up by the recipient cells, exosomes undergo one of three fates. They either disintegrate to release internal components, fuse with lysosome for degradation, or go back into the extracellular fluid. Current platform technologies for therapeutic exosomes can be categorized into three. First, naive exosomes. Naive exosomes are naturally produced exosomes from the originator cells, such as stem cells or immune cells. Their exact pharmacological component and mechanisms are poorly understood. Second is a direct cargo loading method. Direct cargo loading method give ex vitro stimulations on isolated naive exosomes to load small-sized cargos. These methods allow specific targeting of biological molecules that treat diseases, but it is limited on stability, productivity, and size of cargo to be loaded. Lastly, the most advanced method is an indirect cargo loading method. By engineering exosome producing cells, macromolecules could be loaded into the exosomes during the natural exosome production. This addresses some of the concern around the stability and cargo size with direct cargo loading method. Currently, this technique is not only used for cargo loading but also for targeting specific tissues and organs. We also summarize the research trend of several human cells in terms of genetic modification, the productivity of exosomes, scalability of cell culture, and safety. Mesenchymal stem cells and neural stem cells can produce therapeutic exosomes having low immunogenicity. However, stem cells are limited in large-scale cell culture. HEK293 cells have many technical advantages, such as easy manipulation and high scalability, so they are being used to develop engineered exosome delivering therapeutic cargo, but there may be potential tumorigenic issues. As dendritic cells are antigen-presenting immune cells, their exosomes also can induce an antitumor immune response. Therefore, dendritic cells are commonly used to produce therapeutic exosomes for cancer vaccine. Cardiosphere-derived cells, amniotic cells, and CAR T cells are being used or considered as therapeutic exosome-producing cells due to their low immunogenicity and therapeutic effect. The exosomes produced with various cells are actively being studied as therapeutic options to numerous incurable diseases, such as Alzheimer's disease, cancer, neurological disease, and heart disease. More detailed information on exosomes and their therapeutic potential can be found in our review paper. Thank you for your attention.